Today's preparation determines tomorrow's achievement. If you are a CSIR NET aspirant and if you are planning to revise unit number 7, System Physiology in Animals, then this video is for you. So today I'm going to talk about tips and techniques to revise unit 7, System Physiology in Animals better. So with no more delay, let's get started to the video. Hey ardent learners, this is Caroline Green from Biotechnica. If you are belonging to zoology or belonging to medicine, and I'm going to tell you that this unit number 7, System Physiology in Animal, is a great chunk for you to prepare for CSIR NET Life Sciences. So let's dive into the session. Okay, we already know about Unit 7 System Physiology in Animals syllabus. So I'm going to take you to the syllabus and let's talk in detail about it. So we know that there's going to be blood and circulation. So we have to understand what is this blood, what is the composition of the blood, and what are the blood corpuscles that are present, what is hematopoiesis, how does this blood are actually formed, and what are the formed elements. And we also have to understand the function of the plasma and what's the difference between the plasma and the serum, and how much is the blood volume actually present, what happens if the blood volume is increased, and what happens if the blood volume is decreased, how this blood volume is actually regulated in our body, what are the blood groups. So we'll be talking about ABO blood group, RH blood group, all these things in detail. Along with that, we have to understand about the hemoglobin, and we will be also talking about the immunity and homeostasis in detail in this unit. The next one is cardiovascular system. After understanding about blood, we will be talking about cardiovascular system in case of humans. Here we'll be talking about the anatomy of the heart, and we'll also be calling our heart as myogenic heart, very important. So you have to study what is this myogenic heart. Without an external stimulus, even the heart can undergo contraction of auricles and ventricles. And what are the specialized tissues that are responsible for the contraction of auricles and ventricles? You'll be studying about SA node and AV node, everything in detail. And then we'll be talking about the electrocardiogram and electrocardiograph and what's the main principle behind it. What's the most important thing about this ECG? Very important one. And usually we'll get a lot of questions in ECG. The next one is going to be cardiac cycle. Okay, how much time does it take for the entire heart to go in for a complete contraction and relaxation? We know it's going to be 0.8 seconds. This is also another important chunk in this unit, we can say. And heart is going to behave like a pump, and what happens to the blood pressure, and how this actually heart is going to contract and relax. Is there any neuronal control in it, and how they are regulating it? Everything we are going to study in this unit. The next one is the respiratory system. So this respiratory system, we'll be talking about different species. So not only about human beings, how is the respiratory system in case of all other lower organisms. And we'll also be studying about the anatomy of this one and how the gases have been transported. Suppose if you're talking about carbon dioxide, how carbon dioxide is transported in the form of carbaminohemoglobin or in the form of bicarbonate ions, or how it is actually been transported. In case of oxygen, how it is transported can be in the form of oxyhemoglobin. So we'll be studying everything in detail. And the next one is exchange of gases. How is these gases are going to be exchanged, and how the waste have been eliminated through the process of respiration, and how respiration has actually been controlled by the neural and the chemical regulations we'll be studying in this one. The next is going to be the nervous system. So we'll be studying about the different types of neurons and the structure of neurons, and we'll be studying about the action potential, and we'll also be studying about the anatomy of the brain, the spinal cord, and we'll also be talking about the central nervous system, peripheral nervous system in detail about this, and very important, we'll also be studying about the neuronal controls, everything in detail. and then sense organs. So we'll be talking about how the eye actually works, how do we hear, and how we respond to all the situations. So this unit talks about the entire human physiology. So if you want to study this in detail, you can go in for it. Suppose if you're not comfortable with this unit, then you can leave, because if you're from botany and if you're not comfortable with zoology, you can leave and study the rest of the units. But if you are going to be a zoology graduate, or if you are belonging to medicine, and this is going to be another scoring unit for you. Okay, the next is going to be the excretory system. So this is in case of human beings, very important. We have to study about kidney. So we have to talk about the kidneys, the type of kidneys, and how the urine is actually been formed. So we'll be studying how the urine is going to be concentrated. Here comes the role of countercurrent mechanism or countercurrent exchangers. So we'll be studying about Henle's loop. We will also be studying about vasa recta, and we'll also be studying about all the nephrons that's actually involved, all the nephrons, juxtamedullary nephrons, cortical nephrons, everything in detail, and how the waste has been eliminated. We are going to study that. And micturition is the process of elimination of urine or ejection of urine, and how water has actually been balanced. So suppose you are in a desert and you have to conserve the water that is present in your body. Suppose if you are drinking a lot of water, then what will happen to the urine? You used to excrete a lot of excess urine with the more amount of water. And how is it going to affect the blood volume and blood pressure? And here comes the role of, we'll be talking about RAS mechanism. We'll be studying about renin, angiotensin, aldosterone mechanisms. So this is very important mechanism that we'll be studying in this one. The next one is going to be thermoregulation, how a body temperature is maintained, how chemicals, neurons, physicals, and acclimatations are going to take place. We are going to study it here. The next is going to be digestive system in case of human beings, how a digestion is actually taking place, and how the food that is broken down into pieces, or we can say the larger macromolecules which are broken down into micromolecules, have been absorbed into the blood. So we'll be studying this in detail, but in excretory chapter, we'll be studying the reabsorption of the materials back again. In digestive system, we'll be studying about the absorption of the material. And here we'll be talking about the basal metabolic rate. And very important is this unit, I would say, because endocrinology is very important topic. When we are going to talk in detail, I'm going to explain all the important topic in this one. I'm explaining this one. So endocrinology is very nice unit where you are going to understand everything. What are the endocrine glands that are actually present, and how the hormone is going to act, where it is synthesized, where it is going to act upon, what are the roles that it has, what is going to happen if they're going to secrete excessively, or hypersecretion or hyposecretion. And then comes the reproductive process, like we can say gametogenesis, which can be spermatogenesis or oogenesis. What are the hormones that are involved in the production of sperm? What are the hormones that are involved in the production of egg? What are the hormones that are involved during menstrual cycle? What are the hormones that are involved during pregnancy? Everything in detail. We'll also be studying about the neuroendocrine regulation in detail. So this is all about the syllabus that we have in CSIR NET Life Sciences, which is unit number 7, System Physiology in Animals. Okay, what are the reference book that you have to follow in this unit? The first one, The Principle of Anatomy and Physiology by Brian Derrickson and Gerald J. Tortora. Yes, this is one of the book which you can follow. The second one is going to be John E. Hall's Guyton and Hall Textbook of Medical Physiology. You can also follow this book. The next one is going to be Shaw's Outline of Human Anatomy and Physiology. So these 3 books you can literally follow for this unit number 7, System Physiology in Animals. Okay, what is the weightage of marks going to be for this chapter? Yes, so part B, usually you will get 2 to 3 questions, so it may be variable, and in part C it can be 3 to 4 questions for this unit. So you can, if you are from zoology or any other background who well aware of all these things, then you can literally read it for a while and you can attempt the questions very easily. Okay, and now let's talk about what are the important topics to be studied from this unit, which is unit 7. different neurotransmitters. So you have to study what are the different neurotransmitters and its action potential. And very important, this neurotransmitters and its action potential always be expected to be combined with another unit, which is unit 2, cellular organization, and also with respect to the membrane system. Ultimately, there's going to be neurotransmitter which will come and bind to the receptors present on the membrane. So it's always correlated with unit number 2. And it's also correlated with unit number 4, which is cell signaling, cell communication and cell signaling. So that is also another important thing. And very important, ECG, electrocardiogram, or we can say electrocardiograph. This is very important, 2 important terminologies. So mostly a lot of questions are usually asked in ECG type of questions, which is very important topic you should never miss. Another one, cardiac cycle. So how does the heart actually undergoes the process of contraction and relaxation, whether it's auricles or ventricle? How much seconds does it takes for one heartbeat? We'll be talking in detail. That's a very important one. The next one is going to be heartbeat. We'll be studying about heartbeat, and which is tachycardia, bradycardia, everything, and blood pressure. So we'll be talking about the hypertension and everything in detail, and blood volume regulations, yes, and different serum contents. So these are some of the important topics that you have to take into consideration. The next one is going to be the endocrine system. So if I have to list out some of the things, we can say first you can concentrate on endocrine system. So in endocrine system, you have to talk about all the hormones. Like we can talk about all the pituitary glands. We can talk about. This is really an important thing, and we'll be also talking about the glands in the pancreas and adrenal glands. Very important thing. So you have to remember all these things. So hormones. And where are they synthesized? Are they coming from the anterior part of the pituitary or posterior part of the pituitary or in the middle lobe? So adenohypophysis, or we can talk about everything in detail. And what's the function of all these hormones? You also need to know this one. So first thing, you can concentrate on endocrine system, and then you can proceed on with the cardiovascular system. And then you can proceed on with the reproductive system. In case of males and females, you can just concentrate on. Yes. Okay. Next is excretory system. So first you can concentrate on endocrine system, and then you can move in for this, we can say you can move in for cardiovascular system and then reproductive system, and then you can move in for the next important topic, which is going to be the excretory system. Here you have to study about how a urine is actually formed. So what are the nephrons that are involved in it, whether it's cortical nephrons or juxtamedullary nephrons, or which is going to be maximum, and what are the structures of these nephrons you have to study. Very important. It's going to be the countercurrent mechanisms, very important one. So this is mainly responsible for the concentration of urine. And then kidney filtrations, you have to study what's the different structures and parts of nephron, as I already told you, and how water balance is actually maintained. So here you will be talking about RAS mechanism, renin-angiotensin aldosterone system, very important one. The next comes the respiratory. So concentrate endocrine, and then you can proceed on with the cardiovascular, and then with the reproductive system, and then you can go in for this excretory system. After that, you can come in for this respiratory system, which is gaseous exchange. And what is the role of hemoglobin? What are they going to do? Are they going to carry oxygen, or what's their role in it? You have to study, and you also have to concentrate on Bohr's effect. And oxygen association or oxygen dissociation curve. That's what it's mentioned, like the behavior with oxygen, like left and right shift. So you have to remember this one. The next is going to be nervous system. Already it has a correlation, I already told you, with the chemical coordination, or we can say neuronal systems or endocrinology. So the same goes over here. And reproduction, yes, I already told you. Male reproductive system and female reproductive systems. What are the hormones that are involved in it in the process of gametogenesis and ovulation. The next comes sense organs. So very specifically, you can concentrate on ear. Yes. And finally, the muscle movement. How does the muscle actually contracts and how the muscle actually relax, sliding filament theory. So what's the role of actins and myosins? What are the hormones that are released during that process? So everything you need to know in detail about this one. Okay, so now we are coming on to the point of what to do during your revision time. So I'm going to tell you the tips and tricks to revise this unit better. What's the first tip? Tabulate all the different neurotransmitters and their action, very importantly, for easy revision. This is very important because I told you the first important thing is going to be endocrinology. You can finish off within a day, within 2 to 3 hours you can finish off. So you can make tabulation like this according to your own interest. You can list out all the neurotransmitters and where are they produced, what is the action. So I'm going to take one example and I'm going to tell you adrenaline. It's called as the fight hormone or the flight hormone. And when does it will be produced? It is produced during stressful situations. Yes. So during that time, it increases your heartbeat. So increases the heartbeat and the blood flow. So everything is actually related to that. So you should remember what is the role of this adrenaline hormone. So there are many hormones like noradrenaline, GABA, acetylcholine. Very important. This has a very important role and it is very important for learning, memory and all these processes. And there are many more neurotransmitters like dopamine, serotonins, endorphins, everything. What's the second tip? Make colorful images. When you're going to study human physiology in detail about it, you should always have colorful images for easy remembrance. If you want to recollect at the last moment, it's always good that you have colorful images, which will be helpful for you to have a very quick revision. So this is about how actually the neurotransmitters are going to be moving from the axonal part of one neuron and it will be going to the dendrites of another neuron. So that is what is given. So let's understand this one in detail. If you're going to see a paragraph, it might be very difficult. So if you have a colorful image, it's going to be very easy. Okay, first, action potential arrives at the axon. So we already know about axon terminal. So let's consider this is going to be one neuron. I'm not writing everything in detail. So there's going to be the dendrites of another one. So let's consider here, we are going to talk this one and this correlation we're going to talk about. So action potential arrives at the axon terminal. So like this, there would be axon potential, which will be developed at the end of this axonal bulb, or we can say axon terminal. And now what's going to happen? The voltage gated calcium channels are going to be open. So you can see over here, since the calcium are maximum at this area, or we can say this area. So this channel is going to be open. So when there's going to be a change in the action potential or there's a creation of action potential along the axonal terminal, this calcium gate channels are going to open and this calcium is going to come in. Yes. So calcium enters the presynaptic neuron. So this is the presynaptic neuron and this is the postsynaptic neuron. So now the calcium has gone inside. So now what the calcium is going to do? The calcium is going to signal the neurotransmitter vesicle. Vesicles are very sac-like structures which used to have the neurotransmitters inside. So as soon as the action potential is generated, calcium goes in, it strikes onto the neurotransmitter vesicles, and now the neurotransmitter is going to break open, or we can say the vesicle comes from this area and moves towards the membrane, and now they are going to release all the neurotransmitter. Let's suppose take this as an acetylcholine. And now this acetylcholine is actually released by the process of exocytosis. Yes. So now this acetylcholine is going to come over here. Now it has to enter to produce a signal to the next neuron cells. So they will go and bind to the receptor. So neurotransmitters are released and they will bind to their specific receptors. So they'll bind to the acetylcholine receptor, and now there is going to be generation of signals over there. And now this postsynaptic cell is also going to receive the signal. So if you're going to study everything detailed through the picture, it's going to be very simple at the last minute revision. Okay. Tip number 3. Create comparison table. This is really important when you are going to talk in case of animal system physiology, because you have to study a lot of things and you have to compare things. So it's very good that you make comparison table. So one example I'm giving over here, how does the muscle actually contract? Or we can say the muscular contraction and how is the relaxation actually takes place. Just now we have done with this one. So action potential is created and calcium usually goes inside, activates the neurotransmitter vesicles, and neurotransmitter vesicle goes towards the membrane, and then they will break open, and through exocytosis all the neurotransmitters like acetylcholine comes over, and this acetylcholine binds to the receptor. Yes, now what's going to happen? Yes, now there's going to be sarcolemma. Yes, sarcolemma are stimulated now, and inside the muscle, suppose if you're talking about this muscle, inside there's going to be sarcoplasmic reticulum, and the endoplasmic reticulum and muscles are called as sarcoplasmic reticulum. As soon as the signals are coming over like this, what would happen is this sarcoplasmic reticulum is actually being stimulated, and now what they'll do, they'll open the calcium channels. And now this calcium channels are going to open, and all the calcium is going to be released into sarcoplasm, nothing but the cytoplasm of the muscle, which is called as sarcoplasm. And now this calcium is a very important role. We already know about actins and myosins. So usually actins are the one which actually moves and myosin will be in the center. So what exactly happens? Actins are usually masked by some substances. Suppose if I'm drawing something like this, and there's going to be trophonins which are actually present like this. So they used to mask this one. So this is going to be trophonin, and there are going to be trophomyosin which actually surrounds them like this. Trophomyosins. So now usually when there's no muscle contraction, the actin will also be bind by the trophonins or trophomyosins. Now what exactly will happen? Since the calcium is maximum, calcium has more affinity towards this actin comparatively to this trophonin. Now what happens? Actin will, or we can say the calcium will go and bind to the receptors here. So let's take like a star point over here. So instead of this trophonin, now this calcium will go and bind at this region. At this region like this. So instead of trophonin, this will go and bind. They will just unwind, we can say. Now what exactly happens? This trophonin molecules are gone. Trophomyosin is gone. Now the actin is filled by this calcium. And now there occurs the pulling action of this actin. Now the actins will keep moving, and that is why we will say the sarcomere size actually decreases. This is the process of contraction. If you're going to study in a tabulation, it's going to be very simple. And relaxation is very easy. Acetylcholine esterase is going to break the acetylcholine. Yes, and now there's no longer, calcium will not be there. Calcium will go into the sarcoplasmic reticulum. If calcium goes, there is no increase of calcium. So ultimately the trophonin, trophomyosin will be sealed in the actins. So there is no pulling action. So there is no contraction. This will happen during relaxation process. So comparison table is a very important trick that you are going to follow. Okay, what's the next one? Create mind maps for easy interpretation. It is very important that you're going to have a lot of mind maps. So I told you when we talk about the sense organs, so you can follow ear. So I've taken one as an example. So we know there are going to be outer ear, the inner part, the middle ear and the innermost ear. Outer ear, middle and the inner ear. Outer ear, we used to call it auricles, or we can call it pinna. We're not going for detail. And middle ear, we already know after this auditory canal, there's going to be 3 types of ossicles, ear ossicles or bones which are actually present, which is malleus, incus, and stapes. Stape is the most molest bone that we can ever see. And then it proceeds on to the innermost ear. We'll be talking about the cochlea and all, which is the organ of hearing. So if you're going to have a mind map like this, so it will be talking about what's the function of the outermost ear, what's the function of the middle ear, what's the function of the innermost ear. So you can easily understand if you create mind maps when it comes to system physiology in case of animals. The next one, create flowcharts. This you can follow very specifically for reproductive system, because even you can follow it for endocrinology, because where you will be having a lot of hormones. So you can follow it for reproductive system or endocrinology, where you can make it like a flow chart and you can literally follow it. Okay, so here there's one flow chart which is given over here about the follicular phase, which is nothing but the menstrual cycle they are talking about. What happens in follicular phase, what happens in the ovulation phase, what happens in luteal phase, everything in detail. If you're going to see this at the last minute for your revision, it will be really helpful. So create flowcharts, and then it's going to be very easy during your revision time. Next, tip number 6, create flashcards. So here we already know about the cardiac cycle. So we'll be studying about this in detail, which is a very important chunk, we can say, in this unit. So we already know about the auricular systole. Systole means contraction. So auricular systole usually lasts for about 0.1 second. Whereas the right and the left auricles are going to contract for 0.1 second. And then the ventricular systole. So when the ventricles are going to contract, then it lasts for about 0.3 seconds. So during this time, these AV walls, the auricular ventricular walls, or we can say the tricuspid and the bicuspid walls, are going to close. So that will create LUB sound. The next one is going to be joint diastole. The last one is joint diastole, where both the auricles and the ventricles are going to relax for about 0.4 seconds. So totally it's going to be 0.8 seconds. So if you have a flashcards, it's going to be very easy. So create a flashcard. The next one is going to be tip number 7. So tip number 7, create concept charts. So you can avail Konceptika Lite, which is created by Biotechnica. So where you can easily understand this human physiology or system physiology in case of animals very easily. So you will have a lot of pictures like this with a quick notes which will be given over here. So this is another trick that you can literally follow to revise this unit very much easier. This is another one which you can easily see the pulmonary circulation and the systemic circulation. This is also from the Konceptika Lite. The next tip is tip number 8. Give importance to graph related questions. So I told you ECG questions will be maximum. So when you pay attention to graph related questions, then you are going to get good marks. So pay attention to graph related questions in this unit. So one example, you can see it over here. So they are talking about the yes wave of normal human ECG originates from which of these things. So they have given option A, option B, option C, and option D. So this yes phase, we can say, which is in the purple color, you can literally see over here what does it actually represents or originates from. It is mainly the late depolarization of ventricles which are moving towards the AV junction. So the correct answer is going to be this one. Yes, so give importance to graph related questions. Another important tip for this unit is analyze previous year question paper to get the insight, and you will also see match the following questions. So I'm going to take you to some examples of the previous year question papers. Graves' disease is associated with which of these things? So we know Graves' disease actually happens when there is an increased amount of thyroid hormones, or we can say thyroxins. Thyroid hormones. So the answer for this question is very simple: excess of thyroid hormones. So this is an endocrinology question, which is a previous year question paper, very simple one. So you can literally see over. The second question, during urine formation, excretory system, how is the filtration at the glomerulus? So if you're drawing a Bowman's capsule and along with the glomerulus, so we can say, how is the urine is actually formed over here? Is it an active process where ATP is involved, or is it mainly because of osmotic pressure, or it is a pressure dependent physical process, or it is a non-energy mediated transported process? So the answer is pressure, where we'll be talking about the hydrostatic pressure that is seen in the glomerulus and the osmotic pressure, and we'll also be talking about the hydrostatic pressure that is actually in the Bowman's capsule. So it is a pressure dependent one, which is actually a physical process also. So the answer for this question is going to be third one. Okay, the next previous year question paper which will give you an insight is in a healthy kidney excretory system again. So here they have given glomerulus hydrostatic pressure. It's very simple. The pressure that is created when the blood is going to be filtered in the glomerulus. Since it comes from the efferent arterioles, there are some pressures created by the blood that is actually incoming. So they have given, I'm just writing GHP as 75 mm Hg. And the next one is going to be the glomerular capillary colloid osmotic pressure. This is a pressure created by the plasma proteins. So that pressure I'm writing GCO as 40 mmHg. And the next one is the hydrostatic pressure in the Bowman's capsule. Okay, yes, I'm writing hydrostatic pressure in Bowman's capsule. It is going to be 20 mmHg. Now they ask you to find out the effective concentration, or we can say the filtration pressure, or we can say the net pressure. So how we can calculate? Yes, first you have to understand what is the pressure of the blood that is coming inside. So GHP minus of GCO and HPB, which I can write it like 75 minus 40 plus 20. So 75 minus 60 is going to be 15, very simple. So the answer for this is going to be option second, which is 15 mmHg. Tip number 9, analyze previous year question paper, the same one. I'm going to give you another example, inhibin. So we know about inhibin, we know gonadotrophin releasing hormone from the hypothalamus is going to secrete a very important hormone, which is going to be follicle stimulating hormone, and this is going to stimulate the seminiferous tubules, and this seminiferous tubules we'll be talking about Sertoli cells, and this Sertoli cell is going to produce inhibin. And what's the role of this inhibin? If there is maximum secretion of follicle stimulating hormone, they will do feedback mechanism again. Inhibin from Sertoli cells of testis inhibits. Very simple, FSH. So answer is going to be option number 2. Okay. The last one, you can also get insights about match the following question. This is also an ECG question, very important. So you used to get questions like this. So P wave is depolarization of atria, which means the contraction. QRS is the depolarization of ventricles, and T wave is going to be the repolarization of atria, and reduction in the size is coronary ischemia. So this is the next one, which is very simple. So practice previous year question paper. The last tip, tip number 10, make short notes for all important topic. I told you all the important topic for this unit. So make very short notes, which can be in the form of pictures or flashcards, or it can be in the form of flowcharts. A very concise paragraph you can make for everything for your easy revision. And remember the order of blood flow through the heart walls. And you can also go in for mnemonics. Okay, how can I remember? You can have some mnemonics like try performing better always tricuspid, and then it goes to pulmonary, and then to bicuspid, and then to aorta. Yes. The next is mnemonics. I already told you what are the pituitary hormones. So we know there are going to be anterior part of the pituitary, which is called adenohypophysis. And we know there is going to be posterior part of the pituitary. This is anterior part of the pituitary, and this is posterior part of the pituitary, and this is going to be the middle lobe. We already know. And what is this anterior part of the pituitary is going to secrete? What are the hormones? It is based on endocrinology. How can I remember all the hormones? You can just remember flat pig. So F is referring to follicle stimulating hormones, comes from this. Luteinizing hormones, yes. ACTH, adrenocorticotrophin hormones, and thyroid stimulating hormones, which is going to stimulate the thyroid to produce thyroxins, which is going to be T3, T4, and then prolactins and growth hormones. So if you remember flat pig, you will not forget the hormones which are produced from the anterior part of the pituitary, which is going to be adenohypophysis. And we know about the middle lobe of the pituitary is going to produce melanocyte stimulating hormones. This is mainly responsible for the pigmentations. So this is produced only from the middle lobe of the pituitary. And if you have to talk about the posterior part of the pituitary, we already know about ADH, which is vasopressin. This has a correlation with the excretory system. This ADH is responsible for water reabsorption. And this oxytocin is actually produced during the parturition process for the contraction of the uterus to eject the baby fetus outside. Yes. So it is antidiuretic hormone. If a person is going to have very less amount of ADH, then it causes diuresis, where water reabsorption is not going to be happening. So instead, lot of water will go through the urine. This has a very important correlation. This do not comes through the hypophyseal portal system. So you have to remember anterior part is connected to the hypophyseal cell portal systems, but this is not connected to the portal system. Okay, another example for a mnemonic is adrenal gland. Yes, so you should know the adrenal glands are going to secrete some amount of hormones. What will happen if they're going to secrete higher, or we can say hypersecretion and hypersecretions, which is a very important thing that you have to know in this chapter. So one mnemonic is canes. You can remember cortisols, aldosterons, norepinephrines, epinephrines, and all the sex hormones like testosterone, estrogen, progesterone. All this thing you can remember using this mnemonic. Yes. So every preparation, make it a masterpiece. So this unit is going to be a very important chunk for you if you are belonging to zoology, or if you really love zoology or animal physiology, where you can fetch very good amount of marks in this unit. I hope everybody liked this video. If you really like this video, please like, share and subscribe to our channel Biotechnica. Thank you all of you for joining. We'll meet in the next video. Thank you all of you.